Hello there. Can I start? Yes. So very interesting presentation so far. I'm Stefano Monti, Divisional Computational Biomedicine, School of Medicine. And I'm here to present on behalf of a team of investigators that includes also Maria Kuruzinska, Bob Varelas, and a host of others. Our team has several ongoing studies investigating mechanisms driving head and neck cancer, initiation, progression, response to treatment. And for the most part, these studies are focused on the host cancer genome, and transcriptome and epigenome and proteome and so forth. However, today I'll be focusing on a pilot study investigating, among other things, the host microbiome crosstalk and its potential relevance for early disease detection and targeted treatment. Next. So head and neck cancer is more precisely defined as head and neck squamous cell carcinoma. It's quite an aggressive disease with less than 60% 5-year survival rate, and with the majority of cases presenting as tumors of the oral cavity. Of particular interest to this audience is the fact that the oral microbiota is the second largest after the gut in the human body. And that changes to the oral microbiome have been shown to be associated with oral cancer. Next slide. So, excuse me. In our pilot study, we collected close to 80 oral biopsies stratified into groups comprising controls, 3 types of premalignant lesions, and cancer. And we performed so-called total RNA sequencing, which allows us to quantify both the host transcript and microbial communities. And importantly, for the analysis of the sequencing data, we applied a range of bioinformatics tools developed in-house, both by my lab and by Evan Johnson's lab. The main goals were to identify candidate transcriptional biomarkers of early transformation and to look for association between host gene expression and microbial abundance and their relationship to pathway activity. The results were generating many, but one of the highlights of the host transcriptome analysis was an increased immune inflammatory response associated with lesion progression. And so we wanted to investigate this aspect and its relevance to the host microbiome crosstalk. Next. So this is a busy slide, but I want you to take home 2 points. In healthy condition, as shown on the left, standard oral infection, say induced by the mechanical damage of mastication, is kept under check by a well-functioning immune system, mainly regulatory T cells, Tregs. However, under condition of so-called microbial imbalance, dysbiosis, as shown on the right, this can lead to an excess of immune response, so to speak, an excess of Tregs and Th17 cells. And so it can lead to inflammation, which in turn is often associated with cancer. And so given the observation on our data of an increased immune inflammatory response associated with lesion progression, we wanted also to investigate this bacteria immune response interplay. Next. And here are the 2 main take-home points. On top, we show the results of what is called pathway projection analysis, which confirms that indeed inflammation in immune response is significantly associated with lesion progression, as it's measured by the activity of relevant pathway, including interferon gamma, IL-6, IL-10, and so forth. At the bottom, we show the abundance of several microbes previously shown to be associated with oral cancer are indeed positively associated with the activity of those same pathways, those same immune inflammation pathways. Fusobacterium, for example, is an important indicator. So let me say that the sample size of this study is relatively small and the analysis is ongoing, but the results are already promising, and we hope to pursue a larger sample collection and profiling in the near future.